Okay, let's unpack this.
Let's do it.
We are diving deep today, I mean really deep, into that crucial spot where biochemistry meets the clinic.
Right.
And our mission here is all about deduction. We're going to be taking these, detailed patient case histories, the symptoms, the lab results that don't make sense.
All the confusing stuff.
And we're going to trace them all the way back to a single underlying molecular defect.
And that's the ultimate synthesis, isn't it? We're not just reciting pathways from a textbook. We are using these high-yield biochemical principles.
Like what an enzyme actually does, where you find it in the body.
And how it's regulated.
Yeah.
All of that to figure out the cause of these, catastrophic failures. The ability to connect classroom kinetics to a child's actual symptoms is probably the most vital skill you can develop in this field.
Yeah, absolutely. Our source material today really throws us into the deep end. You've got 9 super detailed, open-ended cases covering some really critical metabolic crises. And then there's a synthesis section with questions on things like coagulation, carcinogenesis, and aging. So we've tried to group these fascinating defects by theme to make sense of it all. So let's start with a classic puzzle. Case 1. It's a sickly 5-year-old boy.
Okay.
He has poor growth. He's having these episodes of drowsiness, even coma, and he's got high blood sugar.
It looks like diabetes.
It looks exactly like diabetes. His glucose tolerance test was way off. I mean, his plasma glucose stays high for hours after the glucose load. It screams severe diabetes mellitus.
But, and this is the huge but, the insulin measurements presented this massive contradiction.
Right.
The biological assay, the one that measures how effective the hormone actually is at, stimulating glucose uptake, it showed really low, almost undetectable levels of active insulin.
Both when fasting and after the glucose.
Yeah, both times.
And yet, when they took the same samples and used a radioimmunoassay, the RIA,
A totally different story.
a completely different story. That test showed huge amounts of insulin-related material, a massive jump after the glucose load. So how do you square that? High total insulin material, but basically zero active insulin.
Well, you have to look upstream. You have to look at how insulin is made.
At the synthesis.
Exactly. It starts as a single chain, proinsulin, which then has to be cleaved very precisely to give you the 2 active chains, the A and B chains.
Which are then linked together.
Right. And the crucial insight here is that the RIA, it's actually measuring all of those related peptides, proinsulin, the active insulin, and the intermediates. It can't tell the difference.
While the biological assay only recognizes that fully processed active structure.
So if your RIA total is high, but your biologically active fraction is low, there's only one explanation.
The patient is churning out a huge amount of an inactive precursor.
Yes. The failure isn't in the insulin gene itself. It's in the processing. The machinery that's supposed to cleave proinsulin into active insulin is broken. And that precursor molecule can't signal, which is why you get diabetes-like symptoms, even with a sky-high total insulin count.
Wow. Okay, so moving to case 9, we're sticking with glucose failure, but a different kind.
Maturity-Onset Diabetes of the Young, or MODY.
Specifically the type with dominant inheritance. And here, the whole story is about enzyme kinetics acting as a regulatory switch.
It's all about the enzymes.
So we have 2 enzymes that do that first step of glucose metabolism, right? The phosphorylation. You've got hexokinase, which is basically everywhere. It has a super low kinase, meaning it's working at full throttle even when glucose is low.
Got a high affinity system, always on.
And then you have glucokinase, and this one is only in the liver and pancreatic beta cells.
And this is where it gets really interesting. Because glucokinase has an exceptionally high kinetis for glucose, maybe, 10 to 20 times higher than hexokinase?
Which at first sounds really inefficient, doesn't it? Why would you want a poor enzyme to be your main glucose sensor?
Because that high column is its genius.
Okay.
I mean, think about it. Affinity is so low, it's barely active at normal fasting glucose levels. Its activity only starts to ramp up almost proportionally when glucose concentrations get high, like after a meal.
So it's not just an on-off switch. Its reaction rate is actually reporting the glucose concentration.
Precisely. It reports that concentration directly to the beta cell, and that's the trigger for insulin release.
And the experimental data from the source backs this up completely.
Oh, absolutely. When researchers blocked glucose phosphorylation, insulin secretion just stopped. Dead.
And in the MODY patients, when they sustained high glucose levels, their insulin response was just blunted.
Their beta cells just couldn't see the sugar. They couldn't respond properly.
And the genetics ties it all together.
Yep. Common mutations right in the glucokinase gene. It impairs that fundamental sensing mechanism, and that leads directly to the insufficient and delayed insulin release that you see in LODY.
Yeah.
It's just a beautiful illustration of how a subtle change in enzyme affinity can break an entire physiological system.
Okay, so from glucose sensing, let's plunge into the high acid, high crisis world of organic acidemias.
Yeah, failures of the body's catabolic machinery.
Let's start with case 8. It's a 4-year-old girl with these recurrent crises, vomiting, rapid breathing, and a severe metabolic crash. Her blood pH was, terrifyingly low, 6.89.
And her initial labs, they showed a huge spike in lactate, pyruvate, and ketones. Just all the alarm bells ringing.
So they analyzed her liver mitochondria, and the defect was, well, it was obvious.
Pyruvate carboxylase activity was almost zero, just gone compared to the controls, and that single failure explains the whole crisis.
So what does that mean for the patient when pyruvate carboxylase fails?
Well, that enzyme is essential for what we call the anaplerotic pathway. It converts pyruvate into oxaloacetate, or OAA.
And OAA is that critical 4-carbon molecule that has to combine with acetyl-CoA to get the TCA cycle started.
Exactly. So if OAA levels drop because you can't replenish it, the TCA cycle just starves.
It can't process any of the acetyl-CoA coming in.
Right. So pyruvate has nowhere to go. Its only other option, really, is to get shunted into lactate.
And that causes the severe life-threatening lactic acidosis. It's a total failure to feed the central energy cycle.
Catastrophic.
Okay, so that's a failure right at the hub of metabolism. Let's look at a trio of disorders that are about failures to break down amino acids. Case 5, propionic acidemia. The key finding was that when they fed this patient the keto acids of certain essential amino acids, like isoleucine.
She immediately started building up propionic acid, methylcitrate, propionyl-carnitine.
All these specific metabolites.
And those metabolites are the perfect molecular fingerprint for a deficiency in propionyl-CoA carboxylase. That's the enzyme that's supposed to process the propionyl-CoA that comes from breaking down those amino acids.
So the buildup proves the block.
Yep, it's a direct confirmation.
Now, case 6, HMG-CoA lase deficiency. This one has a really striking presentation.
This one is fascinating.
The patient had severe hypoglycemia, but critically, their plasma ketones were completely undetectable during a fast.
And the mother mentioned this peculiar cat-like odor on the child's breath after high-protein meals.
Which is such a specific clinical clue.
It is. And that odor, plus the buildup of very specific acids, 3-hydroxy-3-methylglutaric acid, for one, it confirms the block is at HMG-CoA lase.
But why is the absence of ketones so diagnostic here?
Because that enzyme is essential for the final step of making ketones from the amino acid leucine. So if your body can't make ketones when you're fasting, it's forced to rely completely on glucose.
Which leads directly to that severe hypoglycemia.
Exactly. Even though the patient's glucagon and insulin levels were perfectly normal, the fuel source was just gone.
Okay, and the last one in this group, case 7, showed a really widespread metabolic failure. A young child with a severe skin rash, hair loss.
Yeah, signs that really make you think of biotin deficiency.
Right. And on top of that, they're excreting multiple different organic acids, including propionate and others.
That combination, the systemic symptoms, plus a whole basket of different organic acids, it just screams failed cofactor.
It has to be something that affects multiple enzymes.
And that's biotin. Biotin is a required cofactor for a bunch of carboxylases, including pyruvate carboxylase and propionyl-CoA carboxylase.
So the key finding here was that the patient was excreting a lot of biotin.
Which means they couldn't recycle the biotin from peptides. This wasn't a dietary problem. It was a defect in biotin metabolism.
And the treatment is just a perfect example of biochemical manipulation.
It's beautiful. You just overwhelm the system. You give them massive, superphysiologic doses of biotin.
Enough to get around that broken recycling pathway and supply the cofactor that all those failing enzymes need.
And it corrected the functional deficiency. A really elegant solution.
So when these major catabolic pathways fail, it obviously puts a huge stress on the body's defense systems.
Which brings us to the red blood cell.
Exactly. Cases 3 and 4, we're talking about acute hemolytic attacks. The red blood cells are literally bursting.
And they're triggered by oxidizing agents. Certain drugs like primokine infections or even fava beans.
Right. All these things produce hydrogen peroxide and other oxygen radicals.
Nasty stuff.
Yeah.
So the erythrocyte has to have a defense system, and that system is glutathione, or GSH.
It's the antioxidant.
It's the main antioxidant. It's the sacrifices itself. It gets oxidized to GSSG, and in the process, it neutralizes those dangerous reactive oxygen species.
But that's not the end of the story. You have to recycle it.
You have to get it back. The GSSG has to be reduced back to active GSH by an enzyme, glutathione reductase.
And that recycling reaction absolutely requires NADPH.
It's the source of the reducing power. And you have to ask, since red blood cells don't have mitochondria, where on earth do they get their NADPH?
And in the experiments in the source material, it confirmed it very clearly.
The sole source of NADPH in the red blood cell is the pentose phosphate pathway,
Catalyzed by G6PD, Glucose-6-Phosphate Dehydrogenase.
So when the cell is stressed by an oxidizing agent, the activity of that pathway shoots up to churn out more NADPH for defense.
So if you have a G6PD deficiency, you have low NADPH.
Which means you can't recycle your glutathione.
And your red blood cells get damaged, they lyse, and you get acute anemia. It's a vulnerability that only shows up when you're challenged.
And what's really fascinating here is the heterogeneity. It's not just one disease.
We see two totally distinct molecular defects causing the same clinical problem.
Right. Case 3, the African-American male, he had an enzyme that was thermally unstable. It just fell apart at higher temperatures and had low activity.
Whereas case 4, the Maltese boy, his enzyme was thermally stable. The problem was different.
His enzyme had a very high kilauem for its substrate, NADP plus CABA. It couldn't bind it efficiently, so even though the protein was stable, its activity was still very low.
It's just a powerful reminder that G6PD deficiency isn't one thing. It's a whole family of different molecular errors that all lead to the same oxidative vulnerability.
All right, let's transition now and try to synthesize some of the high-level concepts from the assessment questions. We're moving from these single enzyme defects to more systems-level regulation.
Starting with coagulation, cancer, and aging.
Let's start with hemostasis. Coagulation is this incredibly complex machine.
You really need to know the components of the 3 core enzyme complexes, and they all depend heavily on calcium ions.
Right, the calcium acts like a bridge to the phospholipid membranes.
Okay, so first you have the extrinsic SACE complex, which kind of kicks things off. It needs factor AIA and tissue factor.
And then the intrinsic SACE complex amplifies it, using factors IXA and AIA.
And both of those complexes are designed to activate one thing, factor X.
Which then forms the final machine, prothrombinase, with factor VO. And its job is to convert factor 2 prothrombin into active thrombin.
And clinically, this is so important. Think about warfarin, a really common anticoagulant. How does it work?
It works by blocking vitamin K recycling. And vitamin K is essential for adding these special GLA or gamma-carboxyglutamate residues onto prothrombin and other clotting factors.
So without those GLA residues,
those factors can't bind to the calcium phospholipid complex. They're basically useless.
And on the flip side, if a patient has a dangerous clot, like during a heart attack, we can give TPA.
Tissue plasminogen activator. It enhances fibrinolysis, the body's natural clot-busting process.
Okay, moving to oncogenesis, there's a fundamental difference between 2 types of genetic defects here.
Right, you have oncogenes like MYC and RAS. These are gain-of-function mutations.
They're like a stuck accelerator pedal.
Good analogy. And since they're usually dominant, you only need a mutation in one copy of the gene to start driving uncontrolled proliferation.
In contrast, you have tumor suppressor genes. The famous ones are RB and P53.
And these are loss-of-function mutations. They're the brakes. So to get cancer, you need to lose both copies of the gene. The whole braking system has to fail.
And they have very precise jobs. P53 is called the guardian of the genome.
It responds to DNA damage, its concentration goes up, and it activates genes that delay the cell cycle to allow time for repair.
Then RB.
RB physically binds to a transcription factor called E2F, and by doing that, it regulates that crucial transition from the G1 phase into the DNA synthesis S phase.
We also touch on the Warburg effect. This is the observation that tumor cells, well, they seem to prefer rapid anaerobic glycolysis, even when there's plenty of oxygen.
It's a metabolic shift. It's partly linked to a specific isozyme, PK2, which seems to favor shunting glucose metabolites toward building blocks for biomass.
Like lipids and amino acids.
Right, instead of just maximizing ATP production. And we should also note, as it's in the source material, that a compound called dichloroacetate, which has been studied for anti-cancer activity, is an inhibitor of pyruvate carboxylase.
Okay, our final topic, aging and damage control.
The mitochondrial hypothesis is a big one. It basically posits that reactive oxygen species, or ROS.
The byproducts from the electron transport chain.
Exactly. They cause continuous damage to mitochondrial DNA. And a critical point is that mitochondria don't have the same robust DNA repair capacity that the nucleus does.
And then there are telomeres.
The protective caps on our chromosomes, they naturally get shorter every time a cell divides because of limitations in replicating the lagging strand of DNA.
And the enzyme that fixes this, telomerase.
It's usually only highly active in stem cells, but unfortunately it often gets reactivated in cancer cells, which is part of what gives them their immortality.
And just a final concept here. Why does the cell invest so much more in DNA repair than, say, protein repair?
It's about permanence. A mutation in DNA is a permanent change to the blueprint. It gets passed down to all succeeding generations of cells.
Whereas proteins
Proteins are constantly being made and broken down, they turn over. So a damaged protein is a temporary problem, but a damaged gene is a permanent one.
You know, this whole deep dive has really shown how symptoms like coma or severe acidosis or anemia, they're almost always traceable back to a single critical enzyme defect.
We've seen it all. Failures in hormone processing, the starvation of the entire central energy cycle, the loss of protective reducing power from a G6PD variant.
And we've established how these specific clinical signs, like the absence of ketones during a fast or the excretion of a whole bunch of organic acids at once, they act as these perfect molecular fingerprints.
They let you trace the defect all the way back to its source, deep inside our central metabolism.
And as you think about all this material, here's a final provocative thought for you. Almost all of the therapies we talked about, avoiding fava beans, giving massive doses of biotin to force a reaction, using bicarbonate to correct acidosis, none of them are really cures.
No, they're not. They are fundamentally brilliant biochemical manipulations. You're just compensating for or bypassing a flawed pathway to try and restore some kind of metabolic balance.
It really highlights the immense power and the precision of using targeted biochemical intervention in patient care.
It absolutely does.
Thank you so much for joining us for this rigorous exploration of clinical biochemistry and for using your knowledge with us to solve these really complex clinical puzzles.